Hello, my name is Menahem Moshelion. I'm a plant physiologist studying whole plant-water relations at the Hebrew University of Jerusalem, the Faculty of Agriculture. Today, I would like to share some of the problems and questions I'm facing in my lab regarding the abiotic impact on croplands' productivity, in particular protein crops. I will also describe a functional screening system we designed here at the faculty that enable us to screen for drought-tolerant crops. Well, all crops produce food for us, and the basic mechanism is photosynthesis, where plants use light, water, and CO2 in order to produce the primary metabolite, which is sugar. These sugar are then being transformed into metabolite, all the other metabolites we know, like protein, fatty acid, DNA, RNA, and more. And basically we can say that these metabolites are our food. Plants are autotrophic organisms, which means they use the light, water, and nutrient from the soil, and the photosynthetic process is being produced here at the leaf. The plant need to get CO2 from the air, but at the same time, when they get the CO2 via the open stomata, they lose a lot of water. In fact, the ratio between water loss and carbon gain is 500 to 1 in average. This ratio is called water use efficiency. And the result of this bad water use efficiency we have in crop could see very easily by looking at the water consumption of plants. For example, one maize ear actually need to be irrigated in 100 liter. If you look on avocado, for example, avocado on average need to be irrigated about 500 liters in order to produce 1 kilo of avocado. This very big water use is going to be very harmful under drought conditions. When you look at this graph, you can see in orange here the yield losses in response to drought in all major crops. The losses are going from 60 to 80% in one season. This is a major loss due to drought. This is only part of the problem. The big problem is that we need to improve our food production toward the future. In fact, in 2050, we have to produce at least 50 to 70% more yield than we produce today. That means that we have to improve our breeding processes and going from this blue point to this red one. Breeding processes are very efficient. In the last decades, we can see very nice improvement in all of the cereals and the legumes, and we can hope that this breeding will improve also in the coming years. But please note that cereals has much better improvement rate than legumes. Now, cereals are major carbohydrate sources, while legumes are major protein sources. And obviously protein is a major element in our diet. Breeding for drought tolerant is a very long process. The literature talked about 20 to 25 years per single crop. So, breeding may not be the ultimate goal. I would like today to discuss a different approach, which is different land use. Looking at this figure, we can see global land use for agriculture. What we see here is that today, about 17% of all of the agricultural lands are used to produce human food directly coming from plants. Another 13% is plant-based food aimed for animal feed. And 70% of the lands today are used to produce animal-based proteins. What do I mean by that? If we take all the protein which are animal-based, fish, milk, eggs, and so on, we found that about 50%, 47% is purely beef or mutton. That means that about 50% of our lands are used to produce meat, and 25% are used to produce all the other animal-based proteins, and about 28% are used for all other plant-based diet. we may think about different way of using this 47%. What do I mean by that? When we grow meat on this part, we basically grow proteins. Meat is our major protein source, but in order to produce 100 grams of protein from meat, we need about 185 square meters. But while to get 100 grams of protein from plant, we only need 3.5 meters. That means plant-based proteins are 50 times more efficient than animal plant-based proteins. For that reason, I decided to try and look for legume that is able to grow in semi-arid places like the chickpea, and selecting from this plant the lines that could survive drought the best. I turned to Professor Shachal Abu, an expert in chickpea, and he was kind enough to provide me with 110 worldwide chickpea, and we decided to select them based on functional phenotyping screening. I would like to explain. When you look for a plant that behaves the best under drought scenarios, you can go to imaging or physiological screening or biochemical screening. Physiological screening actually phenotyping the plant performance, while imaging is actually phenotyping the plant morphology. Nevertheless, looking for plant morphology might be misleading. I would like to explain. Think that we take this plant and expose it to drought, and then we screen for many plants under this drought and find that some plants look pretty nice, but some look pretty poor. In fact, these nice looking plants are misleading, because the fact they look nice, it's only because they close the stomata, stop to produce, and start to survive, while we are looking for plants that keep working at least for some time during the drought. Let me give you an analogy. Let's say you are looking for a hard-working worker. Now you come to the field and you look on the worker's hand. Which worker you think work harder? The one with clean hands or the one with dirty hands? The one with dirty hands probably work harder. So the same thing with plants. The plant that work harder usually doesn't look as good, but they produce much more, and many times these are the plants that need to be selected, but if you just go with the visual aids, you may lose them. So we turn to physiological based screening. At this process, we actually do a plant performance analysis, and we look for plants that perform the best under drought conditions. Again, let me give you some analogy. Think about that I want to measure someone's heartbeats throughout the day. I would probably use some sensor like this Apple Watch that measure continuously its response. But if I want also to stress the human being during that time, I have to have another machine that stresses the human being, and then while stressing it and measure its physiological response, I may get some very interesting stress profile. So we do the same, only we do it for plants. We have a system that each plant could be stressed by a specific controller that regulates the irrigation system, and at the same time measure the plant physiological performance. We don't do it for one plant, we literally do it to hundreds of plants, arranged in a randomized design, so each plant gets a different treatment. We have many repetitions for each treatment, and we measure simultaneously the root, the shoot, and the atmosphere. And because we measure everything continuously and simultaneously, we can compare all the plant behavior under normal condition, drought condition, and recovery conditions, while all the plants are exposed to similar environmental conditions. This system, built by Plant-DiTech, an Israeli agtech company, is actually enabling differential treatments, in particular drought, to each plant in the array, and a series of key physiological traits like stomatal conductance, transpiration, biomass, water use efficiency, root fluxes, and more. So we can measure all these traits while exposing the plant to drought. This is a picture of my greenhouse with hundreds of chickpea exposed to drought, while some of them are under controlled conditions. And the screening process is based on 5 stages. The first one, we check before drought start. You see here the system show you that the irrigation is well. Here we look for plant with fast growing and high transpiration rate. So we call it pre-stress cumulative transpiration, and we check for the plant with the highest growth rate and transpiration rate. The second stage is the cumulative transpiration throughout the growth period. The third stage is we check for the dry shoot biomass at the end of the experiment. The fourth stage is we measure the water use efficiency. And the fifth stage, one of the most important part, is we check the resilience. What you see here is drought exposure of the plants. This is reduction in soil water content. So all the plants reduce transpiration, but here at this point we recover the plants and we measure the rate of recovery. The faster the plant recover, the better is the better is the trait for drought response. Taking these 5 characteristics, we screen literally hundreds of plants looking for all of these characteristics, and we can point out plants which actually get the highest mark in all the 5 traits. And the nice thing here that all of them compared one another under the same light, temperature, humidity, and of course soil water content, which is controlled by the Plant-DiTech system. So, in conclusion, what we recommend is to screen for plants. We suggest protein plants, but it could be done to any crops in a continuous and simultaneous manner under different environmental conditions. It could be drought, salinity, or other abiotic stresses. This way, this functional phenotyping system allows us to do a large quantities of many kinds of crop and with high capacity. This enables us to pinpoint the plant which show a better resilience, a better growth, and better in every one of the 5 characteristics I show you. So in the end, when we look again on the map of the global lands, what I think would be wise is to take part of this meat growing lands and turn it into chickpea growing plants. As you remember, the chickpea needs 50 times less land than the meat growing lands and still produce the same amount of proteins. In this way we can keep much part of the meat but also replace big parts of the proteins. I would like to thank you for listening to me. I would like to thank Dr. Ahan Da'al, my colleague that helped me a lot in the chickpea research, and of course the funding agency, the Israeli Science Foundation, the Ministry of Agriculture and Rural Development, the Ministry of Innovation and Science Technology, and the FGF from Germany. Thank you.